Welcome to the evolution of microbial life. Let me get my pointer set up here. Kind of at evolution. Specifically, we're looking at microbial life. That is, things that we cannot see with the naked eye. We'll try and do this. We're going to ask life, and we're going to look at some insights. And that is basically one of the things that we're trying to do right now is create life in the laboratory. And there's a number of things that we can do on this. Just taking mycoplasma, and we're taking the mycoplasma and we're inserting the genome into it, and we've kind of done stuff like that, but we've not made totally life. And these are kind of things that are a little odd. These are living things. Craig Vayner hopes to create an artificial genome and transport the genome into the host-free cell. And we've kind of done some of this, and there's a number of reasons why we want to do this. We would like to produce microorganisms that would get rid of our toxic waste. We'd like to produce microorganisms that will produce our fuels, and basically we'd like to produce organisms that exist under conditions that we don't necessarily know yet. So, but again, we haven't been able to do that. Basically, what I want to do is talk a little bit about kind of episodes in the history of life. The Earth was roughly about 4 billion years ago. I think life first evolved about 3.5 billion years ago. We saw production of oxygen roughly about 2.7 billion years ago. These microorganisms lived alone for roughly about 2 billion years. Microorganisms are in great abundance today. Now, this is kind of hard to understand because, again, we don't have fossilized bones from these organisms because they don't have any bones. They're very small, and so we were looking at kind of different things. Single-celled eukaryotes, we think, first evolved about 2.1 billion years ago. And eukaryotes are organisms that basically membrane-bound organelles. Multicellular eukaryotes first evolved about and again before it was basically prokaryotes. Before this, single. Okay, and so again, here's the origin of the Earth. 4,500 million years ago. So that's a billion years ago. The oldest known prokaryotic fossils are from roughly about 3.5 billion years ago. And, probably some ancestor there common to everything today was present then. You see the production of atmospheric oxygen due to prokaryotes, and then we see production of this coming lineage we have. And we have three major of the bacteria. We have what are called the archaea, and then we have the eukaryotes. And if we go back and we see stuff, so it's down here. Here are the prokaryotes and the archaea. Okay. All major phyla of animals evolved at the end of what we call the Cambrian explosion, which began about 540 million years ago. And so we're not in the billions anymore, you guys. And this lasted about 10 million years. We have plants and fungi first colonized land about 500 million years ago. And we followed by the amphibians that evolved from the fish. If we were to use a clock analogy, ticking down of the major events in the history of life on Earth, we would see kind of something a lot like this. Prokaryotes by themselves for about 2 billion. Eukaryotes going on here. What's that's roughly here? Began Earth because we weren't there. We don't know how it happened. But this is it rises existing. If this, how can organisms first arrive? From the time of the ancient Greeks until the 19th century, it was commonly believed that we had what we called spontaneous generation. That doesn't happen today. Most biologists think that the possibility of life on Earth is simple cells by chemical and physical processes. It was the conditions of the first Earth that allowed us to do this. And basically what we thought we saw here is that we had a reducing atmosphere. And in fact, there was no oxygen present. And this is kind of a big factor. And so oxidation is what kind of degrades many of the molecules that we see in life. Without oxidation, those molecules would last for a long time. In order to get biogenesis, we need a four-stage hypothesis. We basically see there's a chemical evolution in four stages that we see. The first stage, orvolife, is most extensively studied by scientists in the laboratory. It's relatively easy to do. And a guy by the name of Urey, who was a graduate student, in an experiment they built early Earth, in which they took hydrogen gas, water vapor, they sparked it, and then they let it go. And again, here's Stanley Miller and Howard Urey. Basically, what it is, and this, in the absence happens, is that these will develop polymers. If we sample this, we could basically, so what he did is he produced amino acids, several sugars doing this. What happened once you make a bunch of these monomers need to get together, and this can happen on hot sand, on clay, and on rocks that can act as polymers. In stage 3, formation of pre-cells. Now, what can happen what happens is that we take these apopathic molecules, fatty acids, etc., and they will produce a bilayer. That's and then membrane is pretty stable structure. And then secondly, of a gene, and here's an example: RNA, complementary gene, where we see something that can evolve. From this, we have chemical evolution to Darwinian or organismal evolution. Okay, right. We haven't done it in the laboratory, so we don't know. And so, but again, it's a hypothesis and it's something that we do. So prokaryotes lived and evolved on the Earth for roughly about 2 billion years. And prokaryotes are found wherever there's life. They far outnumber eukaryotes. They can cause disease. They can also be very beneficial to us as well. They do a number of different things. Prokaryotes live deep within the habitats that are too cold or too hot, too acidic, too alkaline. Prokaryotes survive. Deep ocean. These smoke are in there. These organs that live there, and you know what? They probably outnumber the rest of us here. Compared with eukaryotes, prokaryotes are more abundant. They typically are much smaller. Here's the head of a pin, and we see a bunch of bacteria that are on the head of this particular pin. Prokaryotes are ecologically significant. They recycle carbon and other vital chemical elements back between organic matter, the earth, and the atmosphere. They are a very important part of our biogeochemical cycle. Cause about half of all human diseases, and typically they're benign, or mostly they're beneficial. So we tend to think about these prokaryotes as wanting to attack us all the time, but that's not really the case. They could really care less. What defines a prokaryotic cell is it basically lacks a nuclei, they lack internal membrane cells, they have cell walls exterior to their plasma membranes, although not all of them do. And so here's an example of a prokaryote that allows it specific structure. Here's a typical cell structure of a eukaryotic animal cell. Here's a typical structure of a eukaryotic plant cell, and we should know the difference. When we look at prokaryotes, they come in three types: cocci, bacilli, and spirals. So they're balls. They're rod-shaped, which is called spiral, which is the prokaryote. Are regular, and some form cells. Okay, and so here's an example of a giant bacterium. This is one is growing up. Xenobacteria have specialization. All the cells they experience chemo to the ability, which is you can produce endosperms, and so this is an example of a phototrophs obtain their energy. Carbon source. Sure. Molecules, but about basically used fertilizer. Group They include cardiac, for